Hello students, welcome you all to a new video of Sunanda's tutorial. So in this video we will discuss a small part or very important part of unit 1 of your CSIR Net Life Science syllabus. So this important part of unit 1 consists of bioenergetics, glycolysis, oxidative phosphorylation, coupled reaction, group transfer, biological energy transducers. So this unit 1 is a very large unit, consists of many small yet very important parts. So in this video we will discuss these important concepts. So let's start. At first, bioenergetics. In bioenergetics we will discuss laws of thermodynamics. So at first we will discuss about zeroth law of bioenergetics of thermodynamics. So this law states that if two systems where both are in thermal equilibrium with the third system, they are also in thermal equilibrium with each other. So it follows the simple math formula: if A equals to B and B equals to C, then A will be definitely equals to C. If we put a thermometer, okay, let me give you a daily life example. If we put a thermometer in cup A, if we put a thermometer in cup A and it reads 20 degrees Celsius, it shows the temperature of 20 degrees Celsius. Then we remove that thermometer from cup A and we will put it in cup B. And cup B have also 20 degrees Celsius temperature. So the thermometer still reads 20 degrees Celsius. So now cup A is equals to cup B. Cup A and cup B have the same temperature even if they never touch to each other. Okay. So this zeroth law follows the simple math formula: A equals to C. If A equals to C and B equals to C, then A is definitely equals to B. So what are the core concepts? At first, thermal equilibrium. Two objects share heat until no net energy flow between them. They reach a stable balanced state. And third system, act like standard thermometer. If system A and B shows the same reading as thermometer of system C, then A and B share the same temperature, and when A equals to C and B equals to C, then A equal to B. So it follows the simple math rule. So when scientists already named first and second law of thermodynamics, then they realized this even more fundamental rule was missing, because it underpins the other laws. So they went backward and named it as zeroth law. Okay, because it should be discovered before first and second laws of thermodynamics. Okay. So it is the diagrammatic representation of zeroth law of thermodynamics. When A and B are in thermal equilibrium, A and C are in thermal equilibrium, then B and C should be in thermal equilibrium. Next we will discuss about the first law of thermodynamics, and it states that the energy cannot be created or destroyed. It can only change from one form to another. What are the core concepts? The main thing you have to remember is the energy conservation. the total energy of isolated system stays the same. And internal energy, another thing you have to remember about the internal energy, which is expressed as U or E, the total energy stored inside a system. Another thing, heat, which is expressed in Q, energy that moves because of the temperature difference. And work, which is expressed as W, energy that moves when a force acts over a distance. So these are some key concepts, and the mathematical formula which describes this first law is delta U. U means internal energy is equals to Q. Q means heat plus W. What is W? That is work. Okay, so here the delta U is the change in internal energy, Q means the heat added to the system, and W is the work done in the system. Okay, and this law is otherwise called as is the law of conservation of energy. Okay, then the second law of thermodynamics, which states that the total disorder, or more scientifically we can call it entropy, of an isolated system always increases over time. Energy naturally spreads out and becomes less useful, meaning heat moves by itself from hot object to cold objects, but never the reverse without extra work, obviously. So we have to remember that entropy rises, universal disorder or randomness called entropy always goes up in natural process, and direction of it, two things we have to remember. One is increase in entropy always increases, and direction of it, heat always flows naturally from a hot object to cold object, not in other way around. Okay, next the third law. Then the third law, which states that entropy of a perfect crystal approaches to zero as its temperature approaches to absolute zero. Okay, so this law, once again I am telling you, this law states that entropy of a perfect crystal approaches zero as its temperature approaches to absolute zero, which is 0 Kelvin or minus 273.15 degrees Celsius. Okay, so what are the core concepts you have to remember? At first, entropy or disorderness. Second thing, what is absolute zero? The lowest possible theoretical temperature where a system possesses its minimum possible energy. That means it is in its ground state. And what is perfect crystal? A flawless structure where every atom and molecule is identical, perfectly aligned and completely uniform throughout. Okay. So this law, once again I am telling you, this law states that entropy of a perfect crystal approaches zero. Entropy approaches zero when its temperature approaches to absolute zero. This law was first discovered and described by chemist Walther Nernst. This law establishes that at absolute zero all molecular motion stops and perfect crystalline substance achieves complete internal order. Okay, this is the statement. This is about third law of thermodynamics. Okay, and this is the structural representation of the third law. Then what is Gibbs free energy or delta G? It is the amount of useful work a system can do at a set temperature and pressure. So it is the amount of useful work can be done by a system at a particular temperature and pressure. The change in free energy shows if a chemical change happens on its own or not. And the formula is delta G equals to delta H, or change in heat, minus T temperature delta S, that means change in disorder or entropy. When delta G is negative, the process happens by itself spontaneously, as in case of cellular respiration, and the process is called as exergonic reaction when delta G is negative. And endergonic reaction requires an input of non-spontaneous energy. The process needs outside energy to work, and this is non-spontaneous. This is the case in photosynthesis or anabolism process. Sometimes this delta G becomes 0. The system is balanced, that means at equilibrium. Now the cellular energy currency, ATP, adenosine triphosphate. So during the breakdown of adenosine triphosphate or ATP to adenosine diphosphate releases stored chemical energy to drive the cell work. And energy coupling, we will discuss later on about coupling reaction. That what is energy coupling? Now energy coupling, it is the cells couple highly exergonic reaction like ATP breakdown. During ATP breakdown, it releases some energy to power the endergonic process like biosynthesis and active transport. Do you know what is active transport? There are 3 types of transportation which takes place through the biomembranes. One is active transport, one is passive transport, one is facilitated transport. Active transport which require energy, that means it takes place against the energy or against the concentration gradient. And passive transport which takes place along the concentration gradient. And facilitated transport which takes place by using protein channel which helps in the transportation process. So during active transportation, it requires some energy, and that energy will be provided by ATP or ATP breakdown. Okay. So now we will discuss about glycolysis. Okay. Glycolysis takes place in 2 phases. At first, glycolysis is the first common phase of respiration. It takes place in prokaryotes as well as in eukaryotes, and it takes place in the cytoplasm of the cell. It consists of 2 phases, and each phase consists of 5 steps. So all total it is a 10-step reaction. Okay. The first step is called as preparatory phase, and the second phase is called as payoff phase. So the first phase or preparatory phase and energy investment phase. So this phase breaks down glucose, which is a 6-carbon compound, invests 2 ATP molecule to split this hexose sugar into 2 triose phosphate. The step 1 of this phase is phosphorylation, where glucose 6-phosphate is formed. The enzyme which helps in this reaction is hexokinase or glucokinase in liver or pancreas. Here 1 ATP is utilized, and this process is irreversible and requires magnesium ion. Second step is isomerization, when from glucose 6-phosphate fructose 6-phosphate is formed, and the enzyme which help in this process is phosphoglucose isomerase, and this process is reversible. That means fructose 6-phosphate can be reverse back to form glucose 6 phosphate. Next step is phosphorylation. Here fructose 6 phosphate is converted into fructose 1,6 bisphosphate, and the enzyme which help in this process is phosphofructokinase 1, and here again 1 ATP is utilized, the major rate limiting committed step, and this process is also irreversible. What is rate limiting committed step we will discuss in another video, where I will discuss only about glycolysis. Okay. The step 4 is cleavage step, when fructose 1,6 bisphosphate cleave to form dihydroxyacetone phosphate and glyceraldehyde 3 phosphate. Here the enzyme involved is fructose 1,6 bisphosphate aldolase, and this process is again reversible one. Next step is isomerization. Dihydroxyacetone phosphate can be converted into glyceraldehyde 3 phosphate, and the enzyme which help in this process is triose phosphate isomerase. Equilibrium favors dihydroxyacetone phosphate, but removal of glyceraldehyde 3 phosphate pulls the reaction forward. So these are 5 steps of preparatory phase of glycolysis. Here 2 ATPs are utilized. Okay. Next is payoff phase or energy generation phase. Energy is extracted in the form of ATP and NADPH. Because one glucose yields 2,3 carbon molecule, step 6 to 10 occurs twice for glucose. Because so next is the energy payoff phase or generation phase. Energy is generally yielded or extracted in the form of adenosine triphosphate or nicotinamide adenine dinucleotide, and here because one glucose yields 2,3 carbon molecules, step 6 to 10 occur twice for glucose. Okay, at first step 6, oxidation and phosphorylation. Here glyceraldehyde 3 phosphate will be converted into 1,3 bisphosphoglycerate, and the enzyme involved is glyceraldehyde 3 phosphate dehydrogenase. Here this step will produce 2 NADH total, 1 for glyceraldehyde 3 phosphate. Next step 7, here 1,3 bisphosphoglycerate will be converted into 3 phosphoglycerate, and the enzyme which plays important role is phosphoglycerate kinase, which produces 2 ATP total. So in this step, this is very important step, which produces 2 ATPs all total. Then step 8, which is mutase shift, 3 phosphoglycerate will be converted into 2 phosphoglycerate, and the enzyme involved is phosphoglycerate mutase. Step 9, which is a dehydration step, 2 phosphoglycerate will produce phosphoenol pyruvate, and the enzyme involved is enolase. It creates a high energy enol phosphate bond. This step is inhibited by fluoride. This enzyme is inhibited by fluoride. Then the final step, which is substrate level phosphorylation step, where phosphoenol pyruvate will convert into pyruvate, and the enzyme which help in this reaction is pyruvate kinase, and in this step 2 ATPs are released, and this is again irreversible process. So all total in 2 steps 2 ATPs are found, 4 ATPs are released. So all total we get 4 ATPs, but as 2 ATPs are utilized during the preparatory phase, so the net ATP gain is 2 and NADPH 2 NADPH. Next, the final step of respiration, that is oxidative phosphorylation. Phosphorylation means the generation of ATP which takes place in the presence of oxygen. It is the final stage of aerobic cellular respiration actually. So here it takes place. It takes place in the inner mitochondrial membrane. where cells use energy released from the transfer of electrons from one complex to other. Okay, these complexes are arranged in the inner mitochondrial membrane. Here electron transfer takes place from one complex to another along with the pumping of protons from inner mitochondrial membrane to intermembrane space. It creates a gradient that drives the formation of ATP by ATP synthase. Okay, so what are the key components of this process and steps? At first, electron donor, which molecules play as electron donors. So molecules like NADH and FADH plays as electron donor. So they drop off their high energy electrons at the starting of the chain. Then protein complexes pass electrons down the line. This movement releases some energy. Those released energy moves hydrogen ions from inner mitochondrial matrix to the intermembrane space of mitochondria. Okay, so this builds a strong proton gradient because now the concentration of H plus ion is lower in the mitochondrial matrix and higher in the intermembrane space of mitochondria. So finally electron is accepted by O2 at the end. It combines the H plus, it combines with the proton or H plus to form water. And now the time of ATP synthesis or chemiosmotic hypothesis. Due to this proton gradient, protons wants to flow back into the matrix through ATP synthesis. During this movement of proton from intermembrane space to the inner mitochondrial matrix or mitochondrial matrix, ADP is converted into ATP. So adenosine triphosphate is produced via this pathway. What is biological energy transducers? So these are cellular systems or molecules that convert energy from one form to another. Key examples include mitochondria because it can convert nutrient energy to chemical energy during respiration. Similarly, chloroplast can convert light energy to chemical energy during photosynthesis. ATP synthase converting proton gradient into mechanical and chemical energy. So this is called as the biological energy transducer, which can convert a form of energy to another. Okay, so at first the example 1, ATP synthase, which convert electrochemical proton gradient across membranes into chemical energy. Next, electron transport chain complexes. There are 4 complexes present on the inner mitochondrial membrane, and the electron transportation takes place from one complex to another. During that process, proton is transferred from mitochondrial matrix to intermembranal space of mitochondria, and finally that gradient helps to form ATP. Then chlorophyll or photosystem, they capture light energy and convert it to chemical energy during photosynthesis. Another example is myosin, act as chemical transducer by using chemical energy from ATP hydrolysis to produce physical force and muscle contraction. Then bacteriorhodopsin, it functions as light driven proton pump in specific archaea, converting light directly into proton gradient. So these are some examples of biological energy transducers. So next we will discuss about the coupling reaction, coupled reaction. What is coupled reaction? It specifically merge or link energetically unfavorable or energy consuming or endergonic reaction with energetically favorable, that is exergonic reaction, to smoothly drive any cellular process forward. Okay. So for example, the breakdown and formation of adenosine triphosphate. So the formation and breakdown of ATP are coupled because energy released during the ATP breakdown drives energy requiring cellular process. Okay. While the energy from nutrient breakdown drives the ATP formation. ATP formation. This creates a cycle where exergonic and endergonic process are linked by transfer of phosphate group. ATP hydrolysis release energy by removing phosphate group to form ADP and inorganic phosphate. And ATP synthesis stores energy by adding phosphate group to ADP. So the negative free energy change of ATP breakdown powers cellular work like muscle contraction, active transport. The energy from cellular respiration reattaches that phosphate group to ADP to remake ATP. So this is how they are linked with each other. Energy released during ATP breakdown drives other energy requiring process, and while energy from other nutrient breakdown drives the formation of ATP. Okay. So with this I am concluding my lecture video here. So I have touched each and every point I have mentioned in the thumbnail of my YouTube video. So I hope this video will be beneficial for you all, those who are preparing for csirnet life sciences. So please stay tuned. If you like this video, press the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial to get more valuable videos like this. Thank you.